Next major method for screening, it is sequence dependent screening, and one of the method is screening by hybridization. Nucleic acid hybridization, it is the most preferred and commonly used method for the detection of cloned DNA, because this nucleic acid hybridization technique, it is very easy, very rapid, and it can be used to screen larger population of clones. And in addition to that, the cDNA that is not fully sequenced or cloned, it can also be detected, because if full-length cDNA is not there, then its expression product it will not be produced. So here, incomplete cDNA libraries, they can also be screened by using nucleic acid hybridization. A few co-workers like Grunstein and Hognes in 1975, they devised a method to detect in situ DNA cloned fragments. So in situ hybridization was used by Grunstein and Hognes to detect a particular clone from the population of recombinant DNA or from the gene library. So for this nucleic acid hybridization, the host organism that contain our target clone, they may be called as transformant colonies or recombinant colonies of microbes. They can be screened for the presence of target clone. तो इस पर्पस के लिए हमें माइक्रोब्स को एगर प्लेट के ऊपर ग्रो करना पड़ेगा, इनोकुलेट करना पड़ेगा. आफ्टर इनोकुलेशन नाइट्रोसेलुलोज मेम्ब्रेन को हम प्लेस करते हैं ऑन द एगर सरफेस एंड देन इंक्यूबेट करते हैं. And after that, the DNA colonies, rather microbial colonies, they will appear. After the growth of microbial colonies, the colonies are transferred on nitrocellulose membrane. Or in other words, we can say that transformant colonies, they are replica plated on nitrocellulose membrane, and master plate is retained for comparison. Now, what is the basic step of nucleic acid hybridization, which we can call it in situ hybridization? So here, after the replica plating on nitrocellulose membrane, the colonies on the nitrocellulose membrane, they are treated with alkali like sodium hydroxide. It will result in the lysis of the transformant colonies. After lysis, the colonies, they will be lysed, or the DNA it may be denatured, and after that step, the alkali it is neutralized, and the DNA it will bind firmly to the nitrocellulose membrane. After that, the lysed products, they are treated with proteinase K that will degrade the protein, and after that, the membrane it is incubated at 80 degrees Celsius, and that will again firmly bind the DNA with the nitrocellulose membrane, so that after subsequent washing, DNA will not be removed from the membrane because of its high affinity. Now membrane it is bound to the nitrocellulose membrane, and after that, the DNA probe that is complementary to the target DNA, it is being used. So nitrocellulose membrane it is placed in a solution that contain our probe. Now probe it is a oligonucleotide sequence that is complementary to our target DNA, and it is labeled with some radioactive substances. After that, the hybridization it is there, and after hybridization, the target DNA it is detected by radioautography. So this may be the basic step for in situ hybridization. So first, the microbial colonies that are transformant colonies, they are being grown on a suitable agar medium. After growth, The colonies, they are replica plated on nitrocellulose membrane. So membrane, it is placed in contact with microbial colonies. So that colonies will be transferred on the nitrocellulose membrane. Then this membrane, it is lysed by sodium hydroxide. After lysis, the DNA will also be denatured and it will bind with nitrocellulose membrane. After that, it is neutralized, treated with proteinase K to degrade the protein and subjected to washing steps. Now we have pure DNA that is baked at 80 degrees Celsius, so that again it is tightly bound to the nitrocellulose membrane. After that, we will get a DNA print here on the nitrocellulose membrane, and then the hybridization step is performed with radio-labeled probe, and then it is detected with autoradiography. Suppose this is our colony of interest that contain our target clone, and here it is detected, and now from the master plate this colony, it is picked and it is subjected to further screening. So this is the basic outline for in vitro or in situ hybridization. As I told you that after hybridization, DNA fragment, it is detected by autoradiography. Autoradiography, it is a technique in which we use X-ray film to detect our radio-labeled probe, and positive radioautographs, autoradiographs, they are selected and screened further.